Hello. Welcome to next in a series of review clips designed to assist you in preparing for your Biology 3201 public examination. In this clip, we'll be examining the concept of DNA replication. In short, DNA replication is simply making a copy of the DNA molecule. This is a very important process, and without DNA replication, cellular division will be severely impeded. Remember, cellular division, mitosis, maintains a copy of the genetic code. In order for that to happen when cells divide, it is imperative that the DNA make a copy of itself, or replicate. DNA replication is said to be semi-conservative. In short, when DNA is copied, each new molecule created contains one strand of original parental DNA and one strand of new DNA. To illustrate, if we were to take the strand up top to be the original strand, on the left we have dashed lines, on the right solid. Each of these strands become embedded part of the strand to follow. The parental strand, the original parental strand, becomes part of the new DNA molecule, the daughter molecule. New strands are created in accordance with the complementary base pairing. Remember, Garfield the cat, G's always and only pair up align with C's, A's always align with T's, and vice versa. DNA is copied, replicated, through a series of steps. These steps are initiation, elongation, termination, and finally, proofreading and correction. The first of these steps is initiation. What happens here is the DNA molecule will open up, that is, unwind and unzip, and as a result, new nucleotides can be inserted to eventually create two new strands. Initiation happens according to the following. At a specific sequence of nucleotides called the replication origin, enzymes called helicases unzip and open up the DNA molecule, creating replication forks. Next, DNA polymerase, another enzyme, inserts itself in the replication fork and begins to add new nucleotides, one at a time, to create a new complementary strand. You can see a replication fork illustrated here. Helicase initially opens up the molecule, causes it to untwist, causes it to unzip. DNA polymerase, the enzyme, inserts itself and starts forming the addition of new nucleotides. Of significance, note that the enzyme will always repeat from the 3' to the 5' end. This has significance when you look at the bottom strand, which we'll mention shortly. Following initiation, the next step is elongation. And during elongation, the new strand will continue to grow at the replication forks. Elongation also happens according to a series of steps. Initially, at the 3' exposed in the molecule, the leading strand, DNA polymerase will add new nucleotides. This occurs from the 5' to the 3' end only, that being the reference to the new DNA. In order for DNA polymerase to know where to begin, a small piece of RNA, a primer, is laid down by the enzyme RNA primase. This shows the DNA polymerase where to start. Once begun, the addition of nucleotides continues at a steady pace along the leading strand. Now, as DNA polymerase lays down the nucleotides from 3' to 5' direction only, the second strand of nucleotides are laid down the opposite direction of the leading. This occurs when short copies of DNA are made in spurts called Okazaki fragments. This process occurs much slower than the leading strand and is known as the lagging strand. In order for this to happen, RNA primers are needed for each Okazaki fragment. Then another enzyme, DNA ligase, will stitch together the Okazaki fragments to make a complete strand of DNA. You see this illustrated below. The leading strand is synthesized continually from the 5' to 3' end. This occurs without event. As the strand has been laid down from 5' to 3', the lagging strand occurs in a series of stops and starts. Short pieces, called Okazaki fragments, as I mentioned, are formed in sequence. The enzyme DNA ligase stitches these fragments together. Following elongation, we then reach the process called, or the step called, termination. Here, as indicated by the word, this is the stopping of the copying of the DNA molecule. Normally, this happens when replication forks meet each other or at the end of a linear piece of DNA is reached. Of significance is that termination results in strands of DNA that are shorter than the originals. This occurs because at the leading strand, an RNA primer is laid down to allow DNA to start placing new nucleotides. When the RNA primer is removed, there will be a gap at the 5' end of the new strand, and as a result, it ends up being shorter than the original. At the lagging strand, RNA primers are laid down for each Okazaki fragment, and when all of these are removed and the gaps filled, there is a gap at the 5' end of the new DNA that cannot be filled. As a result, this strand will as well be shorter. Now, in each successive replication of DNA, every time the cell divides, the DNA and chromosomes will get shorter and shorter. This presents a problem for eukaryotic cells because our DNA is linear. Prokaryotes avoid this problem as their DNA is circular. As you can see, the leading strand, the 5' end, gets a little shorter as a result of the removal of the RNA primer. The lagging strand has the same sequence. So at the end result, after termination, both the new strands will indeed be shorter than the original. So this is solved because the ends of chromosomes contain specialized areas called telomeres that guard against this. By definition, a telomere are a sequence of highly repetitive nucleotide sequence at the ends of chromosomes. These do not contain coding genes. In humans, telomeres consist of the sequence TTAGGG, which are repeated several thousand times. As termination ends, the telomere ends are cut off, which make them slightly shorter. As more and more DNA replications happen, the length of telomeres shortens, and the shortening of the telomeres directly relates to the death of the cell. As a result, cells and organisms are unable to live forever. It has recently been found that there is an enzyme, telomerase, that is related to keeping telomeres long. This helps to maintain the longevity of a cell. Cancer cells, which divide more often than any other cell in the body, have telomerase in them. This may be related to the reason that cancer cells are so aggressive in their replication patterns. Again, as you can see, chromosomes contain telomeres at the end. After each replication, the telomeres will get shorter and shorter and shorter. The final stage involves proofreading and correction. With proofreading, this is the process whereby DNA polymerase moves along the newly completed DNA strand, looking for mismatched nucleotides and corrects them by inserting the correct nucleotides. This occurs as DNA polymerase moves along the newly formed strand, recognizing whether or not hydrogen bonds are occurring. If it is not occurring, the DNA polymerase recognizes a mismatched set of nucleotides. As a result, the incorrect base will be removed and a proper one inserted. Questions have been posed on public examinations on this material. I present the following for your consideration. The question is, which is a function of DNA polymerase? A. Cleaves and unwinds double helix. While this is part of DNA replication, this is something that a helicase takes care of. B, joins Okazaki fragments together. Again, this is something that occurs with DNA replication. However, this happens through the action of DNA ligase. Synthesize an RNA primer to begin elongation. Again, part of DNA replication. However, this is the role of RNA primase. The correct answer is C, proofreads base pairing. Thank you for your attention. I hope you found this learning clip to be helpful.